Okay, class. So in the last lesson, we talk about the biosynthesis of triglycerol and phospholipids. So in this in this last lesson for lipid biosynthesis topic, we're going to be talking about the biosynthesis of cholesterol. And so overall, as far as with cholesterol, it's an essential molecule in many animals, but it is not required in the in our diet, the mammalian diet. And so how we can create it is essentially using acetate, and then from there we'll end up eventually making isoprene, which will be a key intermediate to finally get into the stage of creating the cholesterol. And so this synthesis of cholesterol happens in four major stages. So in the first stage is trying to get the acetate to form this mevalonate. And so as you can see, as far as the two carbons of this acetate is right here. So that's one acetate. This is a second one that's right here on the other side. And then the third acetate is right here. And that will form this 6-carbon intermediate. And so how do we get to this? So we use acetyl coenzyme, and what it does is we actually take two acetyl coenzyme and react it with each other using the enzyme of acetyl coenzyme acetyl transferase. So it's going to transfer that acetyl group on to the other acetyl group of the acetyl coenzyme. And so that will end up producing as a whole thing an acetyl acetate. And so as you can see, this is one of the acetyl group that's right here, and then the other acetyl group is right here that was combined. And so in order to continue making this malvinate, because remember when we go back to the previous slide, right here is a 6-carbon atom, not a 4-carbon atom. So how do we get to that? So since we have 4 and then we just need 2 more, it just reacts with another acetyl coenzyme, and it binds right here, and you'll see that group attachment right here that is highlighted. It uses an enzyme called HMG coenzyme synthase, and the name comes from the product that is produced, which is beta hydroxy beta methylglutarol coenzyme. So that is what this molecule is. So you see right here is the acetyl group that was there. This other previous acetate group is right here with the 2 carbons, and then what was recently added was from the acetyl coenzyme that was right here. It bound to the carbon that was essentially located here, which is the same carbon as this one. And so this is a condensation type of reaction. And so from there, right now we're still not at the 6-carbon molecule mevalonate. So we got to get rid of this coenzyme A. And so how that happens is you have 2 molecules of NADPH, which donates 2 electrons, and then utilizing the enzyme HMG coenzyme reductase, it ends up releasing the coenzyme A, and then also reduces this carbon that's double bonded to the oxygen to now be a hydroxyl group. So that was stage one, all encompass of stage one as far as major product. So the second major product going into step two is the activated isoprene. And so how do we get from mevalonate to the activated isoprene? So from there, you see you have this hydroxyl group here and this hydroxyl group located here. So mevalonate has two hydroxyl group. We transfer phosphoryl groups to those hydroxyl group. And so this is ATP dependent type of reaction, utilizing mevalonate 5-phosphoryltransferase. And so it transfer a phosphoryl group right here. And then it goes through another transfer of phosphoryl group as well. This is generally a kinase type of enzyme, and now it has two phosphoryl group, which is more considered a pyrophosphoryl group now. And then from there, you still have this lingering hydroxyl group. So now another transfer of a phosphoryl group goes to that hydroxyl group, and then from there, after you have that transfer and all the hydroxyl group are now removed, this phosphoryl group and the carboxyl group is a good leaving group, and so it actually leaves off in the next section and then produces a double bond in this 5 carbon type of structure, and this actually makes your activated isoprene. And so there is two different versions of the isoprene because with the enzyme isomerase, it can go to two different structure where that double bond is switched between different carbon-carbon bonds. So now that you have this activated isoprene, this is actually a key intermediate that actually forms more than just making cholesterol, which cholesterol is used to make vitamin D, bile acid, and steroid hormones, which we talked about more of the steroid hormones in one of the previous lectures in the introduction of lipids. But with the isoprene, the activated isoprene intermediate, it makes so many other essential compounds used in biosynthesis, and they're listed here. You see some vitamins, carotenoids, plant hormones, and so on and so forth. So we still got to get to that structure of the cholesterol, and have that steroid nucleus that I mentioned in previous lectures. And so this next major reaction is having that activated isoprene go and make a squalene. And just notice that this 1, 2, 3, 4, 5 carbon structure is bounded to each other. As you can see here, they kind of broke it down with these red dashes. That's why you can see the 5 carbon structures again. So it's one right here, 2, 3, 4, 5, and then another one here, 1, 2, 3, 4, 5. Then next one here, 1, 2, 3, 4, 5. Again here, 1, 2, 3, 4, and 5. And then here, 1, 2, 3, 4, 5. And this last one, 1, 2, 3, 4, 5. And so overall, you have a total of 1, 2, 3, 4, 5, 6 of the activated isoprene to form this squalene type of structure. So you're starting to see we're getting more closer to the structure of cholesterol, but we're not quite there. You'll see it more in the fourth step. But how do we get from the activated isoprene to this squalene type of structure? And so what we do is we have two other form of the activated isoprene, and they react with one another, having this head to tail type of condensation, where this carbon right here attached to the tail of this activated isoprene. And so that's formulating this carbon structure, which has two activated isoprene type of structures. So you have 10 carbons attached. But remember, we need to get a total of as far as attachment, 6 of them. So we only got 2 right now. So now we're going to add the third one right here. It's another head to tail, where you have this carbon that's more so at the head attaching to the tail of this molecule. And so now we have a total of 15 carbon chain, which is utilizing 3 of the activated isoprene. And so now, okay, so we got 3 in there. We got 3 more to go. So actually, these farnesyl pyrophosphate can react with each other. And so that's why you have 2 of them showcased here to actually finally form the squalene, and it has the same thing, a head to tail type of condensation, condensing, excuse me, type of reaction, where the head is this carbon and attaching to the tail end of this carbon chain. And so there you actually have your squalene type of structure, and where you have a total of 30 carbons. And now for the final major form of getting to our cholesterol product. So you have squalene, and that goes into making cholesterol. So how is this happening? So you have the squalene, and what it does is it actually forms a epoxy group when it interacts with this enzyme, squalene monoxygenase. And then with the oxygen that's helping with making this epoxide, it's also reduced by the NADPH to produce water. So one oxygen is going to water, and the other oxygen is making this epoxide. And so you end up having what's called squalene 2,3 epoxide. From there, it goes through cyclization. And this is what happens within animal cells. So that's why it was highlighted here. It says animals. And it goes through the cyclization of having these double bonds, transferring electrons and forming bonds to have the central structure of linosterol compound, and you see that it's very close to getting to the structure of the final form of cholesterol. And so within these next steps, it's actually 20 steps that goes into it. So they just simplify this as multi-step. But overall, just knowing that the linosterol will be the main intermediate to finally go into the final product of cholesterol. So here are once again the 4 major steps of creating cholesterol. So you have that acetate, which came from that acetyl coenzyme to end up forming the mevalonate, and that is then you create that intermediate, the isoprene. And so it's actually an activated isoprene by having that pyrrole phosphor group that is attached to the isoprene. And then from there, that activated isoprene make the squalene. And then from squalene, you finally have your final form of the cholesterol. And that is the biosynthesis of cholesterol. And that's the end of this lecture.